Hey everyone, this is Caroline with Fiveable here to present to you tonight about biotechnology. Before we get started, just a friendly reminder, obviously these sessions are free now, which is awesome. But if you want replays, if you want all of my materials, if you want my PowerPoint slides, worksheets, etc., got to sign up for Fiveable Plus, which is just $20 until April 1st. So you might as well sign up. All right, so tonight we're talking about biotech. I'm going to show you a few things. It's not a super lengthy topic just because it usually shows up, I mean, as with everything, kind of on the AP exam in tandem with other things. But I do have an example problem for us as well. It's actually from an old AP, so 2007, so before it got redesigned in 2013. But it's still a good, useful practice with biotech. Biotech is really cool as far as being super cool, wow, I just combined a lot of words there, super applicable to current research, right, in the field. So biotech is a huge field right now. If you're interested in this kind of stuff, it's very lucrative and a pretty cool field to jump into. So we'll go through the most commonly shown up things with biotech for AP. I can show you a lab that I'm working on right now with my kids, which is kind of cool, and sometimes they talk about it. It's the Bacterial Transformation Lab. Has anyone done that in their class yet? Bacterial Transformation Lab? Anyone? There's 13 of you. I thought maybe one of you would have done it so far. Well, yeah, Kathleen, you have. Perfect. Okay, good. Then you're going to be a pro at this. If you haven't yet, this is just really good practice. I'm sure you will. It's one of the big ones that most people hit, but it's also kind of complicated one. So that's a good one to go through a couple times. Okay, so biotechnology, what's the purpose? Why do we care about it? Well, the bottom line is we use technology and biology to create things that are useful for us as humans. It's a beautiful thing. So sometimes we're making useful products. Sometimes we're gaining useful information. We use biotech a lot of the times in, like, we'll talk tonight about developmental biology, figuring out how things grow and develop. We make protein product using this, which we talked about very briefly if you tuned into the bacteria and virus podcast. I think that was last week. My weeks are blending together. And basically, we're just harnessing the power of genetic material and making it useful to us, again, in various ways that we'll talk about tonight. Okay, so a couple main things. So I'm going to go through polymerase chain reaction, gel electrophoresis, like, well, just a lot of big words tonight. Bacterial transformation, and which one am I missing? Ooh, oh, restriction enzymes, which are going to be involved in a few of these. Ooh, we have a question? Oh, hello, Dylan. So it says, do things in the news like CRISPR and stuff fall into biotech? Yeah, absolutely. So CRISPR, we can actually talk about briefly as well, because it's really neat, and it is of kind of very frequent news topic right now. So I'm glad you're paying attention. I should actually mention this because we have such a big audience tonight. So you can pretty much guarantee that one of your 8 free responses is going to have to do with a current event in science. So knowing about things like CRISPR and just like keeping half an ear open through all like the political mumbo jumbo for something sciencey that's happening. Like, I'm trying to think, 2 years ago they did one on bees because there's all that research out that we are killing all of the bees in the world and how big of a problem that is. So they always try to do something that's current. So if you know about things that are going on currently, you just have a better chance of understanding the question a little better. So things like CRISPR and stuff like that could show up, and I can certainly talk about it briefly tonight. Okay, cool. Thanks for asking questions, guys. I'm excited. We have such a big audience tonight. All right, so polymerase chain reaction. This is an important one to know about. So, and sorry guys, most of my slides, if you haven't tuned in before, most of my slides are just like pictures and then I talk at you. So I would highly suggest that you have some paper nearby because I'm going to tell you some important things about these processes and what they're used for. Okay, so polymerase chain reaction is what we use to replicate sequences of DNA that we are interested in analyzing further. So polymerase chain reaction is actually going to be the start of a lot of our other processes that we're going to get into. Sorry, my students are trying to talk to me right now. I'm at school. Sometimes I'm at home. So whenever we see a specific region, whether it's a specific gene that we're looking for that we need to amplify, or whether we're trying to determine like paternity, like everyone loves to talk about biotechnology because they watch Jerry Springer and they want to know who the father is. Polymerase chain reaction is the first part of this step. So basically what you need to know is that there's a couple processes going on here. Let me get my pointer out. The first thing is that we're actually going to denature our DNA. So with polymerase chain reaction, we put in a gene of interest, okay, and we have these special machines that are going to heat up and cool down and heat up in cycles. Okay, so what basically starts is we're going to put in the original DNA we're trying to analyze. And then more importantly, even than the original DNA, is the DNA primer that we choose. Okay, so primers usually are RNA, right? If we go back to gene to protein stuff, but we actually throw in DNA primers that are going to anneal to or bind to very particular regions of this DNA. Specifically, they're going to bind right before the gene that we want to amplify. Okay, and that's going to be really, really helpful because that means we're just going to amplify the gene that we want. So let's say we know, because we have the whole human genome mapped, right? So let's say we know that the gene, I was going to write something down and then realize it's going to be a mirror image for you, so I'm going to stop doing that. But the gene that we want starts with, or like right before our gene, it's like ACCTTTCA, right? We can make a primer that will be the complement of that and add that into our DNA mixture, and it will go and find that lovely segment of DNA. And it's big enough that it's not going to be like, it's not just going to be 3 base pairs long, because that would show up many places in a strand of human DNA. But it's big enough that it will bind to just that particular region. That's where DNA polymerase will bind, and that's where, therefore, that is the gene that we will be replicating. Easy breezy, beautiful. Okay, so what happens here, once the hardest part, again, is finding that primer that's going to basically isolate our gene of interest. The rest is just done by machines. So what happens is we denature, we heat up the DNA to a pretty high temperature, 94 to 96 degrees Celsius. So that's right before boiling point, right? And what that does is it splits our DNA open. Then we lower the temperature down to 68 degrees, okay, a nice chilly 68, which allows for the annealing process, which is where our primers find their target DNA sequences. And then lastly, we go into elongation. So we bump it up a tiny bit to 72 degrees, which is going to be the optimal temperature for DNA polymerase then to crank out the segment of DNA that we are looking for. Okay, and we do this over and over and over again. So 1 strand becomes 2 strands, becomes 4 strands, becomes 8 strands, etc. And it's just the segment of DNA that we are looking for. And that's the segment that's getting replicated, which is really, really helpful, especially when we are trying to analyze someone's DNA. So again, paternity, forensics, right? If we're trying to figure out who committed a crime, we can't just take 1 strand of hair and magically have all the answers, even though that's what they make it look like on like CSI. But we can take that 1 strand of hair, we can isolate DNA from it, and then we can amplify it or make many, many copies of segments that are of interest to us using polymerase chain reaction. And now we have enough DNA, right, that we're able to actually visualize it through something like gel electrophoresis, which we'll get to in a second. Kind of cool, right? Oh, Brandon, so glad you got my CoverGirl reference. Appreciate you out there. Okay, cool. So again, major purpose of PCR, polymerase chain reaction, is just going to be to amplify DNA segments of interest. How do we pick the segment of interest? Well, we work really hard to pick a primer that's going to anneal right next to our gene of interest. Then how do we get this to cycle? Well, we throw in all of our important enzymes. We throw in a bunch of DNA bases, and then we regulate the temperature in a way that some genius scientists came up with so that we can get lots and lots and lots and lots and lots and lots of copies of DNA of a particular region, enough that we can actually visualize it with the human eye. Cool. PCR. All right. Next, I want to talk just briefly, and so these are all going to be kind of interwoven. Like you've already seen, we're talking about PCR, but I'm going to tell you about gel electrophoresis in a second. Same is true with restriction enzymes. So these are kind of similar. The same general principle with restriction enzymes and those DNA primers that we were talking about, except for in this case, we actually have different enzymes. So we have Hindi or Hind3, depending on who you talk to, ECO R5, PSTL. All of these we know of. They're enzymes that exist that break apart bonds in DNA in a particular pattern every single time. Okay, so like what Hindi will do is go and find this particular region of DNA, and 10 times out of 10, it will cut it in this particular pattern. So you'll notice that it's doing something kind of unique in that it's actually cutting the sugar phosphate backbone as well as the hydrogen bonds that are keeping our bases connected. And what it leaves us with are these protruding ends that we actually call sticky ends, right? Because they're open and accessible for binding, okay? And we're gonna use these in both gel electrophoresis and then in bacterial transformation, and I'll get to that in a second, because we can cut our gene of interest with this restriction enzyme, and then we can cut another gene with the same enzyme, and those sticky ends, kind of like Velcro, will find one another, right? And we can actually insert different DNA into existing DNA. And again, that will make a lot more sense when we talk about bacterial transformation. And I'll tell you how this is used for gel electrophoresis as well. You just need to know that these enzymes exist. We're going to do a practice-free response. The 2007 free response that I was alluding to earlier involves restriction enzymes that cut at a particular region, so we'll talk about that as well. But you just need to be familiar with the fact that we do have these enzymes that cut at their own unique pattern in DNA, and we can utilize them to assist us with bacterial transformation and with gel electrophoresis, due to the fact that they're cutting, again, at a very specific region, so we can get specific segments of DNA once we've amplified it using PCR. Questions about restriction enzymes? Cool. This is actually going to maybe be a decently quick lecture, so I'll try to, if there's any other topics you guys want to talk about at the end, let me know. Okay. This is the beauty of the redesign also, I should say, because before 2013, you really needed to know every little thing about everything that I'm saying, and now you really don't. You just need to know how it's applicable in a larger way. Okay, so restriction enzymes, a little bit more. Ooh, we have a question. So Aditi, with DNA mapping, do you mean like gene mapping? Is that what you mean? I'm assuming yes, like gene mapping to see if genes are linked or not? And the answer to that would be no, not really. If you mean like mapping out a plasmid DNA, like bacterial DNA, which I'm going to get to at the end of this, so that might answer your question, then yes. So I guess stay tuned. If you mean like mapping a gene and figuring out where genes are, that's more of a genetics thing, with seeing how often those two genes separate from one another during reproduction. That's the best way I can answer that question, but stay tuned at the end of this. That's where we're actually map out a plasmid, which might be what you're talking about. Okay, so to answer Alan's question, which is, so restriction enzymes are used after PCR. Yes, the answer is yes to that. What are we cutting and why do we need to cut it? Stay tuned. So I'll explain why we need to cut for things like gel electrophoresis, and I'll also explain why we need to cut, more importantly, actually right now, which is going to set us up for bacterial transformation, which is putting genes of interest from a different host into a plasmid, which I'll define for you in just a second. And then we can get bacteria and things to make products for us. But more on that in just a second. I know that was like an obscure way of answering your question, but it's only because I know that I'm going to answer it in a few minutes here. And if I don't, you can feel free to ask it again. Okay. Cool. So here's another probably better example of restriction enzymes. This is what we would use in something like bacterial transformation. And so when we talk about that in a little bit, I'll show you. But basically what we can do here, so here's a plasmid. And a plasmid, again, I really wish that, yeah, I'm going to try it. I'm gonna do a little test. I'm going to draw something on my board and it's going to look backwards to you, but I also think it'll be okay. Okay. Welcome to my great. You see my board? Okay, so here's a bacterial cell. Figure out how to make this the best, but it looks really bright. All right, that's slightly better. So here's our bacterial cell. Okay, and remember that bacteria have their own copy of circular DNA in them. Right there. That has all their regular genes in it. Again, if you can't perfectly see that, I'm sorry. I wish I could make the lighting on this thing better, but or just have it not be a mirror image. Okay. But what bacteria can also have are these little circle plasmids, which you can obviously see on the screen, but hopefully also on my board, right? And that's outside of the bacterial chromosome, right? It's its own little circle that will confer a new trait for that individual, okay? Or for that bacterial cell. Okay. The black one. Yeah, that's better. Okay. So these plasmids we're going to utilize in bacterial transformation because they're easily transferable. And there's 3 different ways that that can happen. Okay. But we can also harness the power of bacteria by putting our own genes into a plasmid. So what do I mean by that? Well, if you take a look at here, we have one of those plasmids. So again, this is something that could be found naturally, or we can harness it from a bacteria. But again, it's just a tiny little circle of DNA. It usually contains a gene or 2, and it's outside of the bacterial chromosome, but they still replicate it, so they would still make the protein that it codes for. Okay, and what we can do here is we can cut it with a restriction enzyme. Okay, and so now we have those sticky ends hanging off, right, with a very particular binding pattern. What we can do then is we can take DNA from another organism. So let's say, and I'm kind of jumping ahead of myself, but it's okay. Let's say we want to have bacteria replicate insulin for us, right? And we actually do this. We use bacteria to create human insulin for people with diabetes. Okay, so we would isolate the gene for diabetes, right? Using PCR, we would replicate it, and then we would cut that gene right at either end using the exact same restriction enzyme. So I kind of think of this as like, think of those fun scissors that you had growing up where you could cut in a funky pattern, right? And each one was like its own little puzzle piece, right? So you had one that did zigzags, one that did wavy patterns. Hopefully you guys know what I'm talking about. If you don't, you missed out on a really important part of childhood, but they're super fun, okay? So basically we're cutting the plasmid with one particular puzzle piece scissor, and then we're cutting our insulin gene with the exact same puzzle piece. Right? So each end, the end of the plasmid and the end of our insulin gene should match up. Okay? So when we put them together, and again, this doesn't work 100% of the time, but we don't need it to, and we'll talk about that when we get to bacterial transformation. But when we put them together, they'll actually seal up. And you'll see that our new plasmid not only contains the original genes, but it also contains a gene for insulin. So then if we put this into a bacterial cell, the bacteria would now be producing the protein for insulin, and we could harness that and use it for distribution for people with diabetes. Okay, so that's really the importance of these sticky ends. They just provide a known puzzle piece. So if we cut one piece of DNA with that same puzzle piece as another piece of DNA, those two can then come together because they have the matching ends that will fit together in order to make a new and larger piece of DNA. So hopefully that answers your question. I know we were talking about scissors in the middle there, all sorts of stuff, but it should be helpful to kind of understand why we would use restriction enzymes after we go through PCR, okay? So that's a really perfect setup for bacterial transformation. So let's jump ahead actually past gel electrophoresis and talk a little bit about bacterial transformation, okay? So this is the process that I just kind of discussed. So bacteria, right, they can naturally pick up these little plasmids from the environment. They can actually transfer them from one bacteria to another, which is called conjugation. I'm going to write this in your little chat box. Conjugation is the transfer of genetic material between bacteria cells. This is like their attempt at mating, if you will. Okay, and basically what they can do is they can transfer plasmids through their pili, right, through the little sticky parts on the outside of them, and that allows them to give a bacteria next to them that might not have like antibiotic resistance, give them that gene. So that's a major way that bacteria can actually kind of transmit genetic material in a lateral way, and then pass it on, right? But the other one is transformation. So this happens when DNA is just naturally occurring in their environment. Perhaps the bacteria has died and then degraded a bit, and then there's DNA out there. Or perhaps scientists have added the DNA in. So more often than not, we are going to be the ones that are allowing transformation to occur. So what happens here is that we have our target gene, okay, and we're going to pair that with, so let's say this is the gene for insulin. We're going to pair that into a plasmid that also has a gene for antibiotic resistance, and I'll explain why in a second, okay? So we're going to get our target gene. So we're going to do two things here. We're going to do PCR on the target gene, so we get lots and lots of it. Then we're going to cut it with particular restriction enzymes. Think your fancy scissors, okay? We'll cut now a plasmid that has antibiotic resistance in it with those same fancy scissors. And now we have a beautiful gene going forward that has a promoter region so that the bacteria actually express this thing, right? They actually go through transcription and translation. We have our target insulin gene, and then we have this antibiotic resistance gene, which is going to become really important in just a second, okay? Okay, so through this process then, we add the DNA, right, that we've just made with both the antibiotic resistance gene and the insulin gene to a group of bacteria, oftentimes E. coli. E. coli are really easy bacteria to transform. They're really hardy, okay? So they're usually used in student labs because we make mistakes and it's okay. And basically, we get these guys really chilly. So if you've done this lab before, once you add DNA to the bacteria, you bring them down to almost freezing. So you put them in an ice bath. Okay, and you usually add some sort of positive cation as well. What that does is the positive cation will be attracted to the negative phosphate heads of the phospholipid bilayer, and the cold will start to make that bilayer a little bit leaky. Okay, so the bacteria are ready to take up our new plasmid, right? Because if you think about it, what we want to do is take that bacteria, open it up just enough that we can get new DNA in there, and then seal it shut, all while not killing said bacteria, okay? So the coldness and the positive cations allow kind of our membrane to become leaky enough without obviously rupturing or killing our bacteria. And then what we do is we heat shock them, okay? So we put them into a warm water bath. And what that does is it provides a concentration gradient. So we have a really warm exterior mixed with that really cold interior that we've been chilling on ice. And it's kind of like a concentration gradient where things will just move from high to low, right? So that DNA kind of gets sucked or vacuumed into our bacterial cell, and then we let them cool down again, and they seal back up, and they're good to go. And some of them will have opened up enough to allow the DNA in and then sealed so that they now have the plasmid, and they would be considered a transformed bacteria. Okay, they now have a new piece of DNA that's going to allow them to make a new protein and do something different, okay? The problem is a lot of these bacterial cells will not have been successfully transformed. Because again, what I'm saying is we're opening them up a tiny bit, allowing DNA to come in and closing them back up without them dying, okay? So in this example, 1 in 5 of these cells, and in real life, it's way less because we probably put thousands of cells in our tubes, and my students usually get like 10 to transform, right? A lot of them won't be successful. 1 in a few will be, okay? So remember, the gene that we're actually trying to get here is insulin, right? But we can't tell just by looking at growing bacteria which ones are making insulin and which ones aren't, right? We can't just be like, oh yeah, that's an insulin-producing bacteria right there, okay? What we can do though is tell if a bacteria is capable of living on a plate that has antibiotic in it. So flash backwards, follow with me, to the fact that we paired our insulin gene with a gene for antibiotic resistance. Reason being, if the bacteria successfully gained this plasmid, they also gained the ability to resist some sort of antibiotic, okay? Because again, they can't have gained the insulin gene without have also gaining the gene for antibiotic resistance. So what we can do then is give them or try to grow them on a plate that is full of that antibiotic that they should have resistance to, which means the only bacteria that are growing, the only colonies that form, that's successfully transformed. Antibiotic resistance gene, which means they also have the gene for insulin. So this is selecting for successfully transformed bacteria. And this is a really important concept to understand. So if you have questions, please ask, because this is complicated, but very, very important. Okay, so again, we are conferring this plasmid for a protein of interest, but we're carrying it with a gene for antibiotic resistance so that we can select for it later on. Okay? Then we can take those bacteria and we can force them to turn out the protein that we want, which is insulin, right? So that'd be making that protein. We can isolate that protein and use it for humans or anything else, and it is a beautiful thing, okay? Cool. Questions about bacterial transformation? Yeah, it looks like we have one. Oh, Alan, sorry. I was just like, it took me a second. I feel like my brain is working at half speed this week. I don't know. We had a crazy week last week. We had 2 days off because it was like negative 50 degrees in Chicago. So anyway, so ligation is just, so to ligate means to glue together. So you can think of DNA ligase. So DNA ligase would, like, for instance, come in here and glue together our puzzle piece ends of our DNA. Okay, so that's what ligating means. If you look back into this process, ligating would be gluing together, like could be the process of gluing together our primers or gluing together our strands of DNA. But again, it's just connecting usually the backbone of DNA, gluing those together. That's what's happening here in DNA ligase, is just making sure we don't have breaks in our DNA anymore. Our backbone, our sugar phosphate backbone, is stuck together. Good question. Okay, cool. So that is bacterial transformation in a nutshell. Because it is one of the big 10 labs, it does oftentimes show up in some way, shape, or form on the AP exam. So it's a good one to know about. It's also very, very complex. I didn't do any labs like this until I was in college, and I was a microbiology major, right? So this was like a big portion of my college education, but a zero portion of my high school education, even in my AP Bio class. But then again, I took AP Bio long before the redesign. Okay, so good one to know about. Definitely rewatch if you have questions. Okay. Last major topic that we'll talk about then is gel electrophoresis, which is quite the mouthful. Okay, but basically what we do here, so gel electrophoresis is a way for us to visualize the DNA that we have amplified, like actually visualize it, see the results, and see the different segments of DNA that we have in a certain DNA sample. So again, the process is similar here. We start with PCR, where we're amplifying specific segments of DNA, and then we will often cut them with restriction enzymes to break them into chunks. Okay, and here in gel electrophoresis, it depends on what your end goal is. But let's say you're doing a paternity test, right? And you're breaking your DNA into chunks. You're cutting regions of DNA that you know exist in all humans, right? But they all vary slightly, and they might be different lengths. Okay, so kind of, and I can show you this better in the results when we take a look at the next slide. But kind of what we're doing is we're cutting DNA into chunks. So let's say we have a mom, a child, and 2 potential fathers, right? We're cutting all 4 of their DNAs in chunks. We should see similar chunks between mom and child, right? And then we should see similar chunks between child and actual father, because they share a lot of commonalities in their DNA. We might not know the specific regions that we're cutting apart, but we know that we should see very similar chunks, lengths of DNA between mom and child and between child and father. Okay, and I'll show you that on the next slide when we actually take a look at an example here. Okay, so we're amplifying our DNA. We're cutting it up into pieces using restriction enzymes. And then what we'll do is we'll load it into a gel. And the gel has little micropores in it, okay, that kind of act like a filter If you can picture like a pegboard or something like that, where you have, or like a really, what's an analogy that I'm looking for here? You know, like any sort of pegboard that's going to stop some things from falling through while allowing others to fall through, and the areas just get smaller and smaller. That's kind of what this gel is. So it has little micropores in it that DNA can travel through. And then more importantly, it has this big old positive charge and negative charge on it, okay? So if you didn't know, DNA is innately negatively charged, which is another reason why we kind of want to protect those phosphate groups when we're trying to get DNA into the bacterial cell and bacterial transformation. So DNA is inherently negatively charged. And that's going to be really helpful to us when we're running a gel, because basically we can just throw a positive charge on one end of the gel. So we'll load in all of our samples of DNA. We'll switch this bad boy on, and our DNA, being negative, is going to be repelled from this end and drawn towards this end. So it will start actually moving through, pulling through. Think of that pegboard. My projector just turned off. Pulling through this pegboard, okay, towards the other side. Okay. Now, the interesting part here, and what we can see kind of in step 5, is that different segments of DNA are going to travel different distances. Okay, and you can probably figure this out on your own, but the smaller segments of DNA are going to travel the furthest because they're going to be able to bounce through the gel more quickly, as opposed to the really, really large segments of DNA, the ones that haven't been cut up as much. They're not going to make it very far because they're going to get all tangled up in the pores. Okay, so they'll be much closer to the starting line then. Okay, and what we can do is we can stain this and we can view it, and we can basically detect different band lengths of DNA and compare that with other sources, right? So again, I'm going to show you a paternity example, but we also use this in crime scenes all the time. So we'll take DNA from the crime scene and DNA from maybe a suspect, okay? We'll amplify it, we'll cut it down with these restriction enzymes, and we'll run it through the gel, okay? And if our banding pattern matches up with suspect and crime scene, then we know we have a match. So that's really what they do in real forensics labs. It's like a little bit less exciting maybe than what you see on TV, and it takes a little more time. But once we've amplified and cut down the DNA, if we have matching banding patterns, we have a matching set of DNA because it's been cut in specific locations in order to show us that. So here's an example of a paternity test. And again, I said we have like a mother, a child, and 2 potential fathers, okay? And what we can do, you'll see, we cut the DNA into a number of chunks, and we can see then what we've ran. So we have our smallest sections of DNA down at the bottom. Those are the ones that traveled the furthest, the quickest, and our largest sections at the top. And then we can compare mother to child and child to male 1 versus male 2. Okay, so what you'll notice here, and it's much easier to see on the right-hand side, is that we have some identical banding patterns between mother and child, all the ones that you see in pink, meaning these are segments of DNA that are identical between mom and kid, okay? Then we want to compare, and you can kind of visualize this right over here on the left. We want to compare who has more similarities with the child. And so we have this unique banding pattern on here, kind of 3 bands in a row, kind of a lighter, and a darker. And that's the same between male 1 and the child. We also have a few other matching bands. So these bands in the middle are going to match, being the same length of DNA, and then these bands at the top. Okay, so this would prove to us that this child is a mix of the mother and male 1. And this is, again, oftentimes how they would do something like a paternity test or a crime scene investigation. Okay, questions about that? Alan, you're 100% correct. I like the way you simplified this. So the gel has little baby pores in it, right? So we put the DNA into the top of the gel. We put a charge on it, so that's pulling the DNA through, okay? And you can picture it. I'm like trying to think of another analogy that might be more helpful. Like picture you have to pull something through a huge bin of sand. Right? You're going to be more able to pull something through that's really small, like if you had to pull through like a clothespin, as opposed to having to pull through a soccer ball or something like that, right? It's just going to be harder. It's going to get stuck more often to pull bulkier items through something that's kind of trying to fight against you. So our smaller DNA is going to pull through quicker than our larger DNA, right? And this allows us to actually visualize not only the DNA, because that's what you're seeing. You're seeing a bunch of DNA bunched up that's been amplified using PCR, but also the lengths of those DNA from shortest to longest. And sometimes we can actually quantify that as well. And I can show you that using this example. These are really complex topics also. So if you're like, what in the world? It's definitely worth a rewatch because all of these things are things that like the best microbiologists and scientists in the world are using all the time. So anyway, that's just validating your potential confusion. But then the patterns that you see, are giving us the actual lengths of each DNA segment, and we can use those to compare. So we're either comparing for direct matches and things like crime scene investigations, or we're looking for similarities if we're looking for something like a paternity test. Okay. Cool. So those are the major methods that you need to be familiar with. And they do show up from time to time, usually embedded in other things as well. Oh man, I missed this part with bacterial transformation, so I'll get to that in a second. Okay. So another really cool thing with bacterial transformation, if you did the lab and you used the green fluorescent protein, if you had like glowing bacteria at the end of it, that green fluorescent protein we actually use pretty frequently in human development or other developmental biology. So when you first originated, you were just a single cell that became 2 cells, 4 cells, 8 cells, 16 cells, and then all of a sudden you were trillions of cells and you became you, right? But the way that humans and animals develop is of high interest to developmental biologists. And so what they can do is they can actually take a look at a blastula, which is what we would call that ball of cells that you originate as, and they can take one of them, let's say one of 16 perhaps, and they can add in, using kind of a transformation technique, they can add in a green fluorescent protein that we can also use as a marker to see if bacteria or anything else has been successfully transformed. So let's say we manipulate one of those 16 cells, and then we end up with a pattern like this. So to me, this looks like our green fluorescent protein, whatever cell they manipulated, ended up in major parts of the nervous system. Looks like the spinal cord, perhaps the brain underneath here, and then some of the nerves down the arm. So that gives scientists a clue, okay, of these 16 cells, this is the one that then gets to differentiated and develops into our nervous system. So if we're trying to genetically modify an organism, we're trying to genetically modify the nervous system, that's the cell that we want to focus on. Okay, so this was kind of a fun tidbit. I like to throw those kinds of things in on the side because, I don't know, cool science things are happening all the time. And that glowing frog is so cool. Yes, Alan, there's DNA inside the gel the entire time. That's what those banding patterns are. It's just dyed DNA. That's what you're seeing. Yep. So DNA is always in the gel. Okay, cool. Another cool thing the transformation and kind of development and manipulating this kind of stuff. If you've heard of genetically modified organisms, right, we can use those restriction enzymes. We can use plasmids and things like that in order to modify and add certain traits into, like we have lots of genetically modified crops. So we can add in a trait that makes it like resistant to a certain pesticide using restriction enzymes, and using PCR to kind of amplify that gene of interest and then implement it into a developing embryo or a developing seed or something like that. And then our products will have a new trait. Okay, because remember, anytime we give something new DNA, we're just giving it the blueprint to make a new protein. And that protein is the thing that actually gives us whatever trait we're looking for. So if it's pesticide resistance or antibiotic resistance or something like that, we can use certain segments of DNA, PCR restriction enzymes, in order to add that new fun trait into our crops or something else. Okay, so if you've heard of genetically modified organisms, that's really what they're doing there. They're using these kinds of enzymes in order to provide things with new DNA. Okay. Oh, another example of bacterial transformation. Amazing. So there it is, insulin gene mixed in with a plasmid. Transgenic bacteria grown in culture. We get insulin, it gets injected into someone who needs it, and the rest is history. Cool. Okay, So I just have like one final example, and I'm actually not even going to go through it in a ton of detail. Oh, let me answer Alan's question first. I'm still a bit unsure about what a plasmid is. There's a term for DNA. So Alan, the plasmid is just that little circle of DNA that's outside of the bacterial chromosome. So it's just DNA. It's just a smaller circle of DNA. It's usually like 1,000 to 2,000 kilobases long. So it's a lot smaller than the bacterial DNA. And it's just an easier way for us to basically transmit or add DNA into an organism, usually bacteria, right? So we can take that little teeny circle of DNA, put a gene of interest in, and then implement it into the bacterial cell, and then that bacteria now has a new capability, right? But we're not manipulating the bacterial chromosome. Like we can't really do that because it would be hard for us to take the chromosome out and then put it back in. Right, so we're just manipulating a little teeny tiny circle of DNA that's easily added into the bacteria. So it's just outside of the bacterial chromosome. Okay, so here we go. So this is going to involve gel electrophoresis and restriction enzymes. It's a really complex question. You'll never get something this complex on the AP exam because this was, again, pre-redesign, but it's still kind of a good way to kind of hash out what we just talked about. All right, so a bacterial plasmid is 100 kilobases in length. The plasmid DNA was digested to completion with 2 restriction enzymes and 3 separate treatments, okay? So we broke apart our bacterial plasmid with EcoRI, and when we got that, we got 2 different segments. We got a 70 kilobase segment, and we got a 30 kilobase segment out of that one. When we digested it with HAE3, let's just call it HAE3, that's nice, okay? It broke apart into a 60 kilobase and a 40 kilobase segment, and then when we broke it up with both, we got 40, 30, 20, and 10 kilobase. Okay, so the first question on here is a mapping question. I'm actually going to skip it because you would never, ever have to do this on the AP exam. So that is good news. We're just going to focus on steps B and C. Okay. E, describe how recombinant DNA technology could be used to insert a gene of interest into a bacterium. Describe then how recombinant bacteria could be identified. And then describe how expression of the gene of interest could be ensured. So basically, they're asking you to explain, like, how could we use this recombinant DNA technology to make insulin or something like that? And that's going to touch on everything that we talked about earlier. So I want you to just jot down your initial thoughts. I know this is kind of a lot. You can look back into your notes about what we talked about, especially with a gene like insulin. And then I'll show you how you would have gotten points on a question like this. Just take just like a minute. Just jot down your initial thoughts of how this could happen. All right, so hopefully you were able to kind of pull out some of the things that we talked about earlier. And again, this would be like a more likely way that you would see a question like this as opposed to having to map out anything. So ways that you could have gotten points here. For insertion of the gene of interest, okay, you could have said a lot of different things here to get points. Okay, you could say that you can cut the gene of interest with a restriction enzyme, either from the source or from a plasmid. But if you say that you're cutting the gene of interest with a restriction enzyme, you would get a point. Okay. You also get a point for saying you would use the same restriction enzyme on both the source and the plasmid of interest. So the source would be like the human insulin gene, and the plasmid would be just a piece of circular DNA that you're going to then insert into the bacteria. Okay. You could say that you are then going to ligate the gene of interest with the vector. So the best way to describe this, I should just flip back and explain while we're looking at this. Okay, so you're basically describing this practice, this example. So cutting with a restriction enzyme, 1 point. Cutting with the same restriction enzyme so that we have matching puzzle pieces, 2 points. Saying then you would use DNA ligase or some sort of ligating or annealing process to stick the gene of interest, this blue guy for insulin, an example, my favorite example, with the plasmid, 3 points, okay? And then you could also mention that the sticky ends or the ends of your restriction enzymes that you cut with are complementary, okay? So there's 4 possible points talking about how you can insert a gene of interest, okay? So you're mentioning restriction enzymes, same restriction enzymes, using DNA ligase or some sort of annealing process to stick those two together based on the fact that the ends or the puzzle pieces match, okay? So that's your answer for that first part, talking about how you can insert the gene of interest. Okay, second point, for identifying the recombinant bacteria, the easiest thing to say here would be that it's producing the product that you want, right? Even if you don't know that's possible. So you could be like, we could check to see if the bacteria is producing insulin. Wow, you get a point for that. Or you could say something a little more complex, like we would then grow our bacteria. We would pair our gene of interest with antibiotic resistance or something like that, and we would then grow our bacteria on a selective medium, kind of like we described earlier, so that if it's capable of growing with antibiotic resistance, we know that it has the gene of interest then as well, okay? So any of those would work for describing how we would identify. And then to ensure the expression of the gene of interest, you could get a point here for just mentioning that you would include a promoter or some sort of region that tells the bacteria to start transcription, okay? A promoter could be any sort of thing. You just need, like, you know, in our DNA transcription and translation, you always need AUG to start off the process. Okay, so you need some sort of promoter region that's like, hey, this is where you can start transcription, right? If you don't have that region, there's no way that DNA replication is going to begin, okay? So those are all your possible points there. I can also, or you can also look up, this is just a 2007 AP Bio question, so you can look that up. If you're ever struggling, there's always, like, you can always look up practice AP Bio questions online. There's a lot of them. Again, the ones before 2013 aren't super applicable, like this one, but there are parts of it that are applicable. And if you're ever not sure, you can always ask your teacher. Hopefully they're an expert at this point of what is and isn't required on the new AP exam. Okay, or you can just ask me, because I know I can answer those questions for you. All right, part C, discuss how a specific genetically modified organism might provide a benefit for humans and at the same time pose a threat to a population or an ecosystem. So this is a good question to kind of just think about, because oftentimes the AP exam will include some sort of like ethical question or ask you to answer an ethical question or pose an ethical question. So this one is right up their alley. So there's a lot of different things that you could have said here. Because it's very broad. You need to propose like some way that this is beneficial, right? So genetically modified organism might provide a benefit for humans. Well, we could modify a bacteria to produce insulin for us, and that's beneficial for us. Or we could modify crops to be resistant to some sort of disease that they're frequently getting, so that they're not getting it anymore, and we have better crops, right? Any of those are going to be how some sort of genetically modified organism might benefit us. And then you kind of need to spin it and talk about some sort of threat to another organism, right? So let's say we genetically modify all of our crops so they're all genetically identical, right? And this is actually true of some of our crops. And then some sort of plague comes that wipes out the entire population because we have no genetic diversity. And then wham, bam, you've also thrown in some evolution into your response. And your AP reader will be so impressed with your ability to understand high-level questions. But the cool thing about part C, and you can see this on the rubric, is you can really talk about anything. The threat doesn't even have to be true or real as long as it makes sense, right? So if you're like, well, if bacteria are then producing insulin and there's an organism nearby where insulin is toxic, that would be really detrimental to that population. You don't need to know if another population is actually going to be affected negatively by insulin in the area, but you can say it, and it means you've thought through the question and you get your point. So I always encourage students on questions like this. You don't have to know if something is true in real life. They don't actually really care that you know like how everything in the world works, because none of us know how everything in the world works. They just care that you're thinking critically and you're like, oh yeah, that could be a really, important benefit to humans while also potentially damaging other organisms, because they really just care that you know that our actions can sometimes have negative reactions elsewhere. That kind of makes sense. But again, you're really free to kind of write about and talk about whatever you want for that part to get your points, which is pretty helpful. Okay. All right. The last slide I have on here is just how plasmid uptake and transformation can be determined. Okay, so we've pretty much already talked about this, but we can determine oftentimes if plasmids or transformations have been successful using antibiotics by attaching an antibiotic-resistant gene, using gel electrophoresis by isolating those plasmids, and by a couple other things. So I'm going to keep this in the file of greatness that is all the biology materials. So if you're interested in looking at this further, you got to subscribe to Fiveable Plus, because it will be there waiting for you and I can answer any of your questions about it. But that's really all I have for you tonight. So thanks for staying tuned and listening to some really, really technical science. Again, if you're confused, it's fair. This is very, very challenging stuff. At the same time, don't stress too much, because you just need to know the basics and how we use these to help ourselves and other species. But again, give it a rewatch. Subscribe to Plus so that you can watch rewatches and make a little bit more sense of this. Otherwise, thank you so much for joining. Thanks for bringing so many people out. Next week is my favorite week, all about the immune system. It's my very favorite. Oh my gosh, I love the immune system so much. If you're interested in the human body at all, or you want to be a doctor or a nurse, this is the week for you. It is so freaking cool. I can't wait to tell you more about it. So tune in, bring your friends, and I will see you then. Have a great week, y'all.